and you'll have to like go in person to register.
Shop and Pathogen Genomics, where he and his team use NGS and bioinformatics approaches to investigate public health issues. Please give me one more time for Dr. Anderson.
Now, how do I got it and get it? No, I don't have that screen option. Yeah. Okay. Hold on one second. Let's get it. Fun with Zoom. There we go. Okay, got it. And do you know if they could hear me online with the mic? Is that clear enough? Perfect. Awesome. Okay, thank you very much. Oh, technology. Let's see, where's Zoom again? Sorry about this. I bring this up one more time. Got to get it there first. Okay. Let me show taskbar. And share screen. There we go. Perfect. Supposed to make things easier. Okay. Thanks so much for inviting me. This will be a lot of fun to chat about. It's been a while since I've had this long to actually give a presentation. Usually in public health, you're at these large international meetings and they say, okay, you get like 3 slides in 5 minutes, or you get 10 minutes or 15 minutes. So this one will be kind of fun. I called it Public Health, Pathogen, Entrepreneurship. And hold on one second, more technical. Worked before. Boy, this is fun. Okay, I'll set the advance manually with the mouse rather than the, oh, there it goes. Now it's working. So it's a different audience than I'm used to. Usually it's, entrenched in public health, working with pathogen genomicists, epidemiologists, laboratory directors. This time I get to have a little bit more fun, talk about, how I got to where I am, a little bit about bioinformatics, a little bit about pathogen genomics, and kind of the objectives that I'd like to get is across during this time is, if you're not already an advocate of pathogen genomics in public health, hopefully you will be by the end. Hopefully I'll instill some excitement for pathogen genomics. If you're developing software for pathogen genomics or a similar discipline, hopefully you'll learn a little bit on how to make your software accessible for public health and for general usage. So beyond just the research academic pursuit. And then if you're uncertain as to the direction of your current research, hopefully provide you with some ideas, some encouragement that, everything is still a possibility. I've had a very wide and varied research career to get where I am right now. So for the presentation, I'll give a little bit of introduction. I'm going to talk about public health, introduce some people to the system, how broad it is, and then really narrow in on pathogen genomics, and then talk some about pathogen genomics, that idea of accidental entrepreneurship. So my scientific career in logos, thought this is a good way of having it, is, I earned my PhD in molecular cellular developmental biology from the University of Colorado Boulder, go Buffs. And most of that work was in cell signaling, cell regulation, stem cell differentiation. But during that time, I really got interested in advanced technologies and applying advanced technologies to real world problems. And especially proteomics. That was right around early 2000s when proteomics was really taking off. So I went to Research Triangle Institute as a postdoc and a research scientist, worked on proteomic methods for biomarker development, computational biology methods in proteomics. but very quickly realized I didn't want to be in the clinical space, and that proteomics, in my opinion, wasn't ready for actual clinical applied use, that there still need to be some technology development. But I really fell in love with microorganisms and microbiology and using advanced technologies there. So I said, okay, I want to leave North Carolina. I want to work in energy. I want to work with microbes. So I went to a company called Luca Technologies in Colorado, got back home, or my adopted home. And for 8 years, I spent working at two startup companies, Luca and Cirrus. And this is where I really got to enjoy working on advanced technologies and real world problems, working on biofuels, biochemicals, microbial communities, strain engineering. Really, really exciting. But as it happens when you're in the energy industry, if you are not doing something that's incredibly disruptive, if you're only making incremental gains, kind of the funding dries up. And when the funding dried up, there was an opportunity in 2016 to the Colorado Public Health Lab for their infectious disease pathogen surveillance, bioinformatics. So I decided to take a job at Colorado. And for what I'll talk about, this talk and some other sections, is how I went from 3 years at Colorado and then created Theogen and then work in public health as a private company rather than as a government employee. So I'll give just a couple quick slides about Theogen, just so you know who I am and kind of what we do. And so for the context of the talk, the simplest thing is, our initial mission, and mission is constantly changing, but our initial mission was essentially to transform public health and infectious disease surveillance through innovative implementation of next generation sequencing and bioinformatics technologies. Pretty straightforward. Is how do we revolutionize public health with these new technologies? And, most of us are public health scientists who work at Theogen. We're only 15 people. Let's say 12 of us are technical, 3 more administrative or quality types, predominantly PhD with some masters, most with bioinformatics or kind of genomic epi, master of public health background. And what do we do? What I like to think of is we have this model of build, train, and support for public health. What we want to do is we want to build workflows, we want to build infrastructure, we want to build capabilities for public health labs to use next generation sequencing in their pathogen genomics program. Here's an example. We do this in a very open source manner. I don't believe in doing it any other way. So we have a full suite of tools for TheaPro, TheaUke, TheaCove, TheaMeta. You can guess just from the name as to what they analyze, prokaryotes, eukaryotes. Yes, there was a discussion whether we should use the term prokaryotes, but it just made sense at the time to do that. And then what we build these, but we train people. We train people all over the US. We train people internationally. Since 2021, let's say 36 trainings, bioinformatics, genomic epi, pathogen genomics, 1,300 participants, 20 states, 43 countries. This slide was made a month ago. It said 5 continents. It said 6 in November. Yes, we went to South America last month to Paraguay for training. So we have 6 continents now. And, this is all done virtually or in person or asynchronously. And lastly, we are a digital company. We are virtual. Everybody is all over the world, from Seattle to Atlanta to Kenya, Portugal, UK, out of 15 of us. And we embrace technology. We embrace all the tools of Zoom, Slack, GitHub, Notion, Terra, which I'll talk a lot about later. And a lot of this type of support and virtualization that allows us to have, weekly support hours with California, and by the way, there's going to be a lot of acronyms. If you don't understand the acronym, please ask. And then please interrupt with any questions too. This is a pretty informal group. But, BRR, Bioinformatics Regional Resource, a CDC program for the Western region. This is California Department of Public Health. This is the Northeast region, where we also have Pathogen Genomics Center of Excellence work. We have international office hours for Europe and Africa twice monthly, international Southeast Asia, and this P&I APAC is PulseNet International, Asia Pacific region office hours. So we're really a global company supporting public health globally. Good journey. So how did I get from starting Colorado Department of Health laboratory and then having a company that's supporting public health globally? So first, I'm going to talk a little bit about public health and what public health is. public health can really be summed up in a single sentence if you really want to get to it. And there's a nice quote that said, the mission of public health is to fulfill society's interest in assuring conditions in which people can be healthy. Sounds pretty simple, pretty straightforward. But if you go to, and it was pretty neat, the Mid-Valley, Ohio Public Health Lab, there's a lot of public health labs out there. They had this nice word cloud of public health, and it is broad. Public health is vast. So it deals not only with, infectious disease, pathogen, genomics, but the epidemiology, social policy, economics, training, health equity, genetic diseases, environmental diseases. I mean, it is massive, okay? And usually you have a public health department, and then you usually have a smaller public health lab. But these activities cover the full gamut of trying to make a healthy society. And within the US, and it's very US-centric, these activities are concentrated in different institutions. So most of you are probably familiar with the CDC, just about 72 miles from here by Uber, and World Health Organization. But to me, the most interesting public health happens at the state, regional, and local level. CDC is a very large institution. And a lot of these are just my opinions. I think that they're very siloed in a lot of areas that they work in. They're very research oriented. They're very critical. But what's really neat about the state public health departments is it's kind of where the rubber hits the road. It's where things happen. It's where specimens come in. It's where you're working with the community. And I find it, this area really, really exciting. And what you find at this area is a couple of different, many different areas. Everything from immunization, tobacco control, emergency preparedness, newborn screening, environmental health, injury prevention, chronic disease. But then my two favorite, food safety and infectious disease. To me, there's just about nothing cooler than working in food safety and infectious disease in general. It's just, hopefully I'll show some of this later. It'll be fun. And the other thing is, you have the CDC, the World Health Organization, but say in the United States, you have a very, very large number of public health laboratories where this food safety and infectious diseases occurring. So some states that aren't very popular, like Montana, might just have one public health lab. Some like Colorado will have, a laboratory in Denver area, but they also might have like an FDA laboratory or an agricultural laboratory. Some place like California is going to have, 20 public health labs. They're going to have like 18, 19 county labs. They'll have the state lab. Maybe they'll have an ag lab or something like that. So it's a vast system. There's probably at least 100 public health laboratories of some form or another across the United States. And not only do they have that food safety and infectious disease, but they also have roles in environmental, chemistry, environmental monitoring, water quality, newborn screening. Most of your public health labs will have punch cards with blood spots that every single day, huge packets of blood spots are dropped off at your public health lab to look for genetic diseases, and especially metabolic diseases in newborns. But also emergency management or emergency preparedness, quality management. And the cool thing about public health laboratories is they deal with, like I said, everything. And I could spend this entire hour just talking about crazy stories about rabies and bats coming alive in the office. They were supposed to be dead when they went to dissect them for rabies. Different stories about Legionella at hotels, different foodborne outbreaks with biofilms, and a restaurant being so contaminated that you could find salmonella in the soil outside the restaurant where they dumped the mop buckets. I mean, just crazy. It's so much fun, scientifically. It's really, really neat, but they cover everything. So in being an infectious disease pathogen genomics, it's a great place to really cut your teeth. I kind of think of it as, when doctors and they go to their residency, they want to work like in that inner city emergency room where they're going to see like all these horrific injuries and incidents. I think local public labs are that for, bioinformaticians and infectious disease. Okay. So in my world, when I think about public health, I'm not really focused on, the obesity problems and people's eating habits and smoking and all that stuff. I really like pathogen genomics. I really like infectious disease and public health. And that word cloud This was just taken from a paper from some of the CDC folk. But, thinking about sequences and outbreaks and data and disease and where it was in the genomes and antimicrobial resistance and virulence and relatedness and transmission, and very, very exciting. So, pathogen genomics and public health. This should be, I don't want to describe this one. So when you think about pathogen genomics and public health, there's kind of three main ways that we like to use pathogen genomics. One is the identification, right? If you sequence the genome of an organism, you should know what that organism is. We have lots of databases. We can make quick comparisons. We're going to know from sequencing it, not only is it E. coli or Salmonella, but what serotype, what virulence genes, whether it has Shiga toxin genes, et cetera. So we have that great way of identifying. For characterization, what we're interested in, is there anything special about it? Does it have virulence genes that we haven't seen before? Does it have antimicrobial resistance genes that we don't know about or weren't expecting there? And that's some neat things that actually fall out. Is sometimes you're sequencing for one purpose and you find something else and you're like, oh, didn't realize that was in this organism and actively spreading in the community, but the conditions haven't been right for it actually to present itself. So you have the characterization part of it. But then you also have the phylogenetic analysis, relatedness. And this is a key part in public health, is a lot of times when you sequence an organism, and once again, these organisms that we're looking at in public health labs, they're coming from actual people. They're coming from clinical labs, from doctors. Someone gets sick, they take a stool smear, or they isolate something, and they send it to the public health lab. So we get these specimens when you're in public health. You sequence them, get that characterization, but you could also, look at all the organisms that have been sequenced before and sequenced nationally. And, have we seen this exact pathogen before? Have we seen this exact sequence? Or is there 1,000 SNPs difference from the last time we saw it, meaning they're just completely unrelated? If we have seen it, when did we see it? Where did we see it? Who had it? And from this, if it's kind of the exact genetic makeup, and we've seen it temporally and spatially in the same place, is this an indication that there's active transmission going on? Or was it just a bad burrito, right? With foodborne pathogens, you'll see a bunch of the same organism in a very short period of time in a very localized place. And usually you could find that they all ate at the same restaurant. One of those stories is, yeah, the restaurant kept the lemons in its water underneath the raw meat, so the raw meat dripped on the lemons. They cut the lemons, threw it in your water, and everybody got E. coli. And yeah, another one of those fun things you learn about. So you think if we have sequencers, we have bioinformatics, we can do all this, so it should be kind of problem solved, right? This technology is available. We have lots of bright people. We should just be doing this and learning so much about pathogen genomics and transmission. Well, not exactly. Public health has a lot of challenges and requirements that are unique to public. Yes, they are a laboratory. Yes, you have highly skilled people, but you're working within an ecosystem of state government for a lot of these places. And when you think about state governments and local governments, and you think about the United States, you have 50 to 100 different fiefdoms of IT and data privacy and data security and procurement and all those types of things, which can make it very, very challenging. So there's a few select areas of challenges that I'll address, and then how Theogen genomics kind of evolved out of a lot of these challenges. So the first one, which is relatively solved, thanks to PulseNet and OAMD, and once again, acronyms, public health. PulseNet is the national and international foodborne detection network. I don't know if any of you are old enough to remember the Jack in the Box incident in California, when they had a massive foodborne outbreak in the late 90s, I believe, early 90s. And in response to that, the CDC started PulseNet, and it was a way that you could start analyzing foodborne pathogens outbreaks in the United States, not just at a local level, but internationally or nationally, because our food distribution system is starting to become so vast that a foodborne pathogen might not just show up locally from a restaurant, but you might have 2 cases in Texas and 3 in Minnesota and maybe 5 in California, and you needed a way to gather that data and compare it and identify the source. But PulseNet and OAMD, which is the Office of Advanced Molecular Detection, which has been around for about 10 years at the CDC, made enormous investments in workforce and instrumentation in public health labs nationally for foodborne detection. And so just about I think every single state public health lab in the United States has at least one MiSeq in it for sequencing, Illumina MiSeq. Many of them have multiple MiSeqs, multiple technologies. A lot of them have NextSeqs now, ONT technology. But PulseNet made sure that at least pre-pandemic, they all at least had one MiSeq. And they had a workforce that knew how to sequence, that they could, do the isolation, get the nucleic acid, perform the sequencing, get it on the instrument. For PulseNet, though, they would use a program called Bionumerics, which was their data management bioinformatics platform. So there wasn't a tremendous need for actual public health labs. So where the real challenges start to begin with next generation sequencing is on the bioinformatics side. When you think about the workforce needed, the bioinformaticians, the IT professionals, the data management, right? Governments aren't very good at responding to things quickly. And especially when you have state and local governments, it takes a long time to start creating positions and career tracks and pay scales that are going to attract these type of people to work in a public health laboratory. It's very difficult. Moreover, high performance computing, right? You're usually confined to working within a state IT department. I've told the story a couple times today already that when I was hired by Colorado to lead their infectious disease and bioinformatics, when I showed up, I had a 32-bit computer on my desk and no cloud access. And I was like, what do you want me to do? Like, I can't do anything with this. It took me 4 months to get a computer that was 64-bit, and it took another 8 months to get cloud access. So you have some real challenges in, resources for computing, and a lot of it too, it's almost understandable, right? Colorado, I think, has 100,000 state employees. And at the time that I was at the lab, maybe there was 60 employees, 80 employees at the lab. So the state IT was responsible for 100,000 employees. And they had to worry about somebody from the DMV loading ransomware onto one of the DMV computers and shutting down the entire DMV department. And yes, that did happen. And then they had to essentially wipe clean every single computer at the DMV and reinstall everything. And then you've got Joel over in the lab that's bugging them for cloud access or bugging them for more computers. So they have bigger problems at a large scale than my needs at a public health lab. There's also a lot of challenges along genomic epidemiology. And by this, I mean, once you have your bioinformatics analysis, how do you integrate that into decision-making for public health? How do you integrate it into what you're going to do differently the next day? And this requires integration between microbiologists, bioinformaticians, epidemiologists, data scientists. And so not only do you need this integration across so many different disciplines, but you also have to do this in a very strict regulatory environment, right? So public health is a really, really interesting place that you kind of get a pass on some things that hospitals wouldn't get a pass on, where you could do things under what's called public health regulatory authority and kind of work in a HIPAA-like environment, but the rules are just a little bit different because you're doing things for public health. But you still have lots of regulatory and strict security and data storage requirements, which can make it very difficult to start integrating genomic data into data that is considered HIPAA covered, like personal identifiable information or personal health information. So this gets to the accidental entrepreneurship. And the reason why I call it accidental entrepreneurship is I didn't set out to start a company. A lot of people, when you think about being an entrepreneur or starting a company or doing something, you do it because like you have this overwhelming desire to start a company, to do something. You've got an idea, you want to change the world. With me, I was in a public health lab. I was there from 2016 to 2019. I was making really good strides in Colorado. I was getting lots of funding, holding regional trainings, getting on cloud computing going. But when I looked at the national landscape for pathogen, there really wasn't enough expertise. There wasn't enough resources. There wasn't standardized platforms or tools. And I really wanted to be a part of transforming public health, not only nationally, but globally. Like there was so much that was exciting me about that. And my position in Colorado as a state employee really didn't afford me those opportunities to do those things. My ambitions had completely exceeded my job description. And if you haven't had a government job, a job description is a very important piece of paper. And some people will use it as a weapon. But I decided that there was definitely a need for non-traditional assistance in bioinformatics in public health to overcome a lot of these government challenges. So in some very interesting conversations with family, I quit my job as a government employee in 2019 and started at that time what was known as Pathogen Consulting. So I was one person. I said, I know there is at least 2 public health labs out there that could afford bioinformatician half-time and have that person remote. And I felt that I could make a much greater impact in public health as that outside contributor and working remotely and working for more than one lab than dealing with being a government employee at the state level. And then sometimes you'd rather be lucky than good, as they say. Who would have thought that it would have been timely starting an infectious disease bioinformatics surveillance company in May of 2019, right before the biggest pandemic to hit the world in modern times. But this pandemic really created an acute need for rapid implementation of bioinformatics in public health. Mainly, let's see where the next one is. Okay, so I can back up here a second. So when I started out, I was still kind of doing things the old way. And by the old way, I mean working on the command line, training people at public health labs to work on the command line, doing things much more in traditional Python scripting or installing algorithms. And that doesn't actually work very well in public health. And the thing is, you just don't have enough people that are skilled to do that. They're going to forget how to do it. It doesn't help with the data organization. You have a lot of turnover. It just wasn't working very well. So and then especially when the pandemic started, that model wouldn't work either because you had a lot of labs coming on and they say, we need something right now. And these types of things take time and they take people. And that was, time and people was not something that was available. So one of my first contracts was with Massachusetts Department of Health. I was working with the Broad a lot, learning a lot about workflow management, containerization. After chatting with a few of you today, that's something that I probably could have talked about a lot more in this, but it's something we could talk about in lunch. It's really kind of I'll talk a little bit more about it in a second. But when approaching these public health laboratories, and they knew how to sequence and they needed data right now. So there needed to be a solution that was, first easy to use. We got to get off the command line. We have to have some type of a graphical user interface, something where a bioinformatics analyst, so if you think about bioinformatics with analyst and developer and engineer, public health labs, they don't need the engineers and developers. You could have a small group of developers that can make tools for all the analysts. So you needed a solution that these analysts could use easy. It was easy to implement and get running. It's accessible, and by accessible, meaning that it could cover from a state perspective, you didn't have specialized software installed. Ideally, it would be something you just log into from a web browser, and then you'd have access to a large number of standardized tools. So, that portable, that standardization. And then probably the last two that are really, really important are the scalable and inexpensive. Public health is very bursty, as we like to say. You get a lot of specimens at once and then nothing happens. Then you get a lot of specimens at once and then nothing happens. So it's really useful to have something that's very, very scalable, but then also inexpensive. I say public health and healthcare are different, and they are, in healthcare, you can get a $5,000 test for something. In public health, it better not cost more than $100 to do it. Preferably, it'll cost $5 to $10. So you don't have a lot of money in public health to do a lot of testing. And then, I talked a little bit in the beginning about working in industry. Those 8 years before I went to public health and working in industry were really, really formative in the way that I was approaching science and approaching applied scientists, applied science. One is, you know, you don't need to build it, you could borrow it, right? It doesn't have to be you created it or invented here, especially in bioinformatics. You know, I like to say that bioinformatics is not the field of dreams, right? It's not if you build it, they will come. It's like if you build it, they'll try to build a better one or build another one. So I was not interested in building anything. I was interested in using the tools that are already out there and just borrowing them and applying them to public health. And then also time is your most valuable resource, right? And that gets into building it. You know, I don't have time to build it. I want to get something where I could iterate and move. And this innovator die, innovation requires iteration. It's, you know, you can't, too often certain institutions will, you know, say, okay, we're going to make this great big tool and we've done a needs assessment and we talked to everybody and okay, in 3 years we'll be done with it and we'll share it with everybody. And that's not the way that I was kind of taught in industry how to do things. What I was taught was, you know, this kind of iteration, the way you do something, do it quickly, throw it out there, have it fail, have it break, have them give it back to you, you fix it again, throw it out there, have it break, go back. So this iteration for innovation is really, really important. And then the other thing too is you only need to be good enough to make the right decision. And by this, we mean that, you know, you don't have to have the latest and greatest tool to make the same decision you would with the old tool, right? You just need to know whether two organisms are related or not. You know, in one of the first jobs I had, they told me they only wanted me to be right 80% of the time, which I thought was really, really odd thing to say. But what they should have said was they don't want me to spend the time to go from 80% to 100% right. So if it took me like a week to be 80% right and 10 weeks to be 100%, they want to waste that 9 weeks. They wanted the, you know, my best guess at 80% and then just iterate. And then also, you know, don't be afraid to make decisions. Don't be afraid to be wrong or to fail, or, you know, that's the way you learn. So when it was growing, I mentioned in the beginning that the Massachusetts lab, I started working with the Broad Institute. The Broad Institute had this platform called Terra that they're using for biomedical research. And it's really, really a neat platform that I learned about. And I thought this could be great for public health because it had a nice user portal. You worked at Chrome browser. It sat within Google Cloud. It used workflow management and containerization. It was incredibly scalable, et cetera. You know, it's a platform that was built by the Broad, Verily, Microsoft, and Google. And at the same time I was learning about Terra, there was this great paper that came out on open pathogen genomics. And if none of you or any of you or some of you haven't read this, I highly recommend reading this paper because it'll teach you a lot about pathogen genomics and kind of these ecosystems we work in. But, you know, Ali Black, Duncan McCannell, Thomas Sibley, Trevor Bedford, they came out with this and it really kind of laid out a framework for what a open pathogen genomics ecosystem and what an environment would look for. And it described, you know, portal, registry, pipeline hub, package hub, workflow management, containerization, versioning, audit, valid. And these are all things that are really needed for public health. And these are all very complex topics on their own. But what was neat about this was I could take those concepts, look at Terra and see how well it was fitting there. And, oh, really? That much? Okay. So we'll speed things up a little bit. So what's really neat about this is, you know, in this ecosystem, Terra kind of checked this out with Chrome and DocStore and GitHub and Docker Hub, but it kind of met all these conditions. And what was really neat was, we don't have to go into Terra too much, but, you know, why do you care about Terra? Why do you care about these types of open platforms? Well, with Terra, because we work with the Broad, we can see, they'll give us statistics on how often our workflows are being run. And we know since 2021, at the end of October, our workflows have been run about 6 million times. So like 6 million times somebody has launched a workflow on Terra that, you know, we wrote, that we designed for public health. And we know that a lot of this was viral, but now it's bacterial. And I'm going to skip through something. I'm running out of time. And we know the users, where they're coming from, and trying to think of what I want to jump to here. So I'm going to jump to the most interesting thing, and this is more from a bioinformatics perspective. So I'm going to fly through, This is the Terra web portal. Go in here, a dashboard. We create one for every paper we write so that people have the data and can reproduce things. It's very data centric. You have bring in tools like that. But what I want to get to is kind of the infrastructure. But this is the repository doc store. It sits on top of GitHub. You'd have your repository of workflows to distribute them. There's tons of workflows available. Very easy to use. So this was in place of a live demo, because live demos don't work very well when you give a presentation. But right now, our workflows that are out there for, a whole host of high priority pathogens for bacterial, for viral, for fungal. We just got done with, a large investigation in Nevada with the CRS outbreak that you might have heard from there. Compliance isn't, this is more for a general public health audience, so I'm going to skip through that real quick. But the infrastructure, what's really, really neat about this and the portability, reproducibility is Terra is what I call a BYOB infrastructure, bring your own billing account. So the Broad supports this Terra instance. And as a public health lab, you log into this Terra instance and here's your workspace that sits on top of a bucket for all your data. And when you issue commands inside a Terra workspace, Terra will communicate with Google and their life sciences API and fire up the virtual machines and containers that are needed to do your analysis, pulling from external resources, and then dumping all the data into your storage bucket. So what this does is it allows you to create your tools in a standardized format, make them available on Terra, and anybody can use them anywhere in the world for free. They just have to pay Google for the compute resource they use. And not only that, it's scalable. So we've literally launched jobs with 100,000 specimens and not even thought twice about it and worried about it being a problem. Where we've had a data table, 100,000 SARS-CoV-2 specimens, analyzed, and in a few hours it's done because Terra will go out, it'll talk to the cloud, it'll fire up 100,000 VMs, and it'll just start chugging through the data as resources are available. And it's really nice, and it's because it's in the cloud, you can really extend it. You could bring in other features, dashboarding and all. And this is some stuff where, we've done in California and during the pandemic, SARS-CoV-2. But one thing I just wanted to touch upon real quick was a few published examples of pathogen genomics and public health that we were involved in. The first one was actually the first case of SARS-CoV-2 reinfection in the US. So it should have been the first case globally, except a medRxiv rejected our submission. And so we actually submitted this reinfection case to medRxiv, and they said, no, we don't want to touch it. You need to send it to a peer-reviewed journal. And so in the 2 weeks that it took us to get into infectious diseases, somebody else from, I think, Hong Kong published a paper on the first case of SARS-CoV-2 reinfection. One of those political things. But, it's pretty neat because you get these actual specimens. You can look at timelines of disease and onset and then use bioinformatics to piece together interesting stories. Then we got another paper out of reinfection after only 22 days with 2 different Delta lineages using these tools. These all can be topics for later during lunch or something like that. We worked with California. We built up their whole COVIDnet platform where it was their 18 public health labs, industry partners, academic partners, over 1 million specimens of data into Terra, submitted to GISAID, having their situational awareness, their genetic epi dashboarding. But one of the most exciting things recently was the Candida auris in Southern Nevada and building out workflows to look at transmission networks and C. auris. And not only that, but then to build out the workflows to look at fungal genomics and then actually see in real time mutations for these antifungal drugs, watching these mutations in the targets of those drugs occurring over time within patients, and then actually seeing in those specimens how it's actually conferring resistance to these antifungals. So, like I said, if you want exciting stuff to work on, public health is definitely a neat place to do it. So in conclusion, the pathogen genomics of public health is incredibly rewarding and it's a lot of fun. The technology is changing right now so quickly, but it's allowing scalable, inexpensive, portable solutions. Once again, after talking to a few people this morning, I should have spent more time talking about workflow management and containerization, but feel free to hit me up during other meetings or during lunch and we could talk a lot about that. That's definitely the way that you're going to make your work more accessible to public health institutions or to other fields. And, and this work really impacts daily lives. So there's a follow-up email if anybody wanted to email me. I think this is being recorded, so that this will be on there. And it's lastly acknowledgements. Some of our best partners in public health have been California Department of Health, Nevada. Didn't have too much time to give credit to Association of Public Health Laboratories, but they're a large nonprofit, kind of a sister institution to the CDC. We work a lot with their global health and food safety, Pulse and International groups there. Wyoming Public Health, Massachusetts, the Broad Institute, especially Christine Loreth and Danny Park, CDC, OAMD, LBB, just too many acronyms to go over now with time. Staff B, State Public Health Bioinformaticists, PHAGE, Public Health Alliance for Genomic Epidemiology, and then this is the team at Pathogen. So it's kind of neat to see how few people you could have and how big an impact that, this group, and especially as a subset group that created those initial workflows that are, we have 6 million and counting analyses done up there. So I think that's it. So questions?
Basically, when you were starting your company, well, how was your approach to like now established connections like all over the globe, which is very cool. But how do you establish connections with all these laboratories? Is it from like people you already knew, or how they work with the government?
Yes.
Or
It was actually it was good timing. So when I joined public health, there wasn't a lot of bioinformaticians in public health. And so we were really sought after for training and regional activities. So in my 3 years at Colorado, I probably gave, I think, 24 talks, both at or at regional, national, and international meetings. So I became very well known within public health for being able to perform pathogen genomics. So when I started my own company, I was kind of already known as, on the inside of the public health family, right? I'm not somebody just trying to make a dollar, start a new company. I'm actually really concerned about public health, like working on these problems, had successful programs before, done some neat work, given training. So it was definitely the networks. Very important. So there's a lot of different things. Your foundational bioinformatics skills are pretty much the same across a lot of different disciplines in bioinformatics. What's special about public health is more the way that the bioinformatics is being used, kind of in a production environment more than a research environment, and the way that things have to be standardized and portable across large networks of groups doing that. So that's where I get into the workflow management and containerization are really, really important for this portability and standardization across a number of different sites. As far as whether you have specific pathogen knowledge, well, it's always helpful to have some pathogen specific knowledge. At Pathogen, we have a lot of people that have specialties in different areas, but you end up working on many, many different pathogens. So it's more important just to understand, I think, the general principles of how, bacterial or viral pathogens evolve, and then be able to understand the data in the context of the genetics of those different pathogens. So I don't think you really have to be an expert in any one. It actually helps to know a lot more. Like I said, because at the state level, you are going to cover so many, or county level, you're going to cover so many different pathogens. It helps to be more the jack of all trades. But yeah, more it's the unique challenges in public health are that distribution, ease of use, working in a network, data management type aspects. Yes. Mm-hmm. Yeah. So this is really interesting. So during the pandemic, if we went to a laboratory and they only had people that had, say, worked in bionumerics and were more on the bioinformatics analyst side of things or just beginning, we could have them up and running in like 90 minutes. Literally teach them how to use Terra, use the workspace, load their data in, run the analysis, and pull the data out. If you had any bioinformatics experience at all, it could take hours, right? Because it was doing something entirely different than you were used to doing things, right? You're already ingrained on how bioinformatics applications should work and how working on the command line. And I admit, it took me weeks to understand Terra. I mean, literally, it took me weeks to understand how things were going on. While if we had untrained people and we trained them just on how to use Terra the way Terra does things, it could be done in 90 minutes. And so it was a really funny kind of counterintuitive way of doing things. But it is very point and click. And if you're very SOP driven, it's very easy to be trained on how to run it in SOP driven fashion. And public health is extremely SOP driven. But at the same time that you have this interface, and obviously this wasn't a Terra talk, but if you went in, you can expose every single variable for every container across the entire workflow in that web page. But they're all optional, and we usually tell people don't touch them unless you know what you're doing. Just use the defaults. But, yeah, good question, but yeah, if you have bioinformatics experience, it takes long. Sure. Oh, absolutely. That's kind of, there's a lot of benefits that come with a virtual remote workforce and a few disadvantages, and that's one of them. Fortunately, we do go to a lot of meetings, so we meet up at meetings. I came out here an extra day to meet up with two of the scientists that are right in Decatur. We kind of have, virtual happy hours, kind of water cooler type stuff. And then you just have to make sure you have a lot of activities to just keep people engaged, even in a digital environment. One of those is our question of the day, right? Every day we put a question, and they're usually really geeky, nerdy questions. And then we challenge people, and everybody's interacting and being competitive. I think the first question I asked was, if you had a mole of M&Ms and it covered all the landmass on Earth, how deep would it be in M&Ms, right? And then so usually I have to put a time limit on how much people can spend thinking about the question of the day, but I think it's about 4 kilometers. But it's yeah. So yes, it is challenging, and I think next quarter, the first quarter 2024, we'll probably have an in-person work week. We'll bring everybody into Denver, and we'll all work in the same location for a week to help with that face-to-face. Yeah.
Signed up for it.